I am a brunette, and I do not change color in the winter time. Your dog or your cat will also shed hair, but when they regrow hair, it is the same color that it was before.
But some animals do shed and grow hair of different colors.
So how does that happen? I would love to understand that. Just how can a single individual have two different colors depending on the light or the temperature of the season? We know a lot about the ecology. So why do these animals do it? It's because they need to camouflage. If they're not correctly camouflaged, they will be highly targeted by predators. It's very important to be the right color for survival. And a lot of studies have focused on the mammal species, but not so many people have looked at the grouse or ptarmigan species. So there's 3 species that undergo color change, the willow ptarmigan, the rock ptarmigan, and the white-tailed ptarmigan.
But as it turned out, when Mafalda started researching the ptarmigans, they aren't just white or brown. Some species can have colors in between those extremities.
That's a very good news for the species, and that's what we see in the mammals, that some populations actually do not molt completely to white. They either stay brown year-round or have an intermediate coat-color that allows them to camouflage in environments with less snow. So we're now seeing that that happens also in the bird species, the ptarmigans. So for a geneticist like me, that's the perfect system or natural experiment.
So how does a geneticist like Mafalda study these birds and hares? First of all, she needs access to the animals. One way to do this is by approaching local hunters in the northernmost parts of the world.
I just want to show how hard our field work is.
Okay, you made it look very easy.
Another more accessible way of getting access to these animals is using museum specimens.
Okay, there we have it. All right. All right, so this Norrbotten. Ooh, it's old, 1875. But the idea is that when we put a specimen in there, it should have an even lighting around it. And there's a color standard that we put there. Here, and actually, so this one I haven't used in the analysis itself, but I'm just putting everything in there in case we ever need it. So this actually looks okay, and this actually looks okay, even though it looks very weird. Yeah. Why do I photograph the museum specimens? Actually, we started by not photographing them and just looking at them and saying, okay, this animal is brown, this animal is white. And we soon realized that it is better to not rely on our own judgment, even though it's very good. One of the species I worked at, you could literally put specimens side by side and construct a whole gradient from white to brown. So this is more complicated to just say, to classify into, this or that. So the photographs actually allow us to objectively study color. So we photograph the animals, and then there's these metrics that the camera is able to capture that we can then use to objectively measure color. So instead of saying brown or white, there's decimals, etc. that kind of explain that variation. So that allows us to do justice to the variation that we're seeing, and to find more complexity.
Researchers working with humans or animals in a lab can collect their samples in a controlled environment. For Mafalda, this is a bit more challenging.
And I want to say that it's not easy. Of course, it's quite challenging to do. But I would have to go to the field to collect those same things. So going to the field means I need to bring the equipment with me. Could be all sorts of tools, all types of tools, and something to maybe even freeze samples right in the field, like a doer that has liquid nitrogen. And I have to put that in an airplane and maybe fly somewhere, and then find the animals, figure out what is the best way that I will, capture this animal. I need permits, etc., etc., etc. It's like there's layers, and even then, maybe, which has happened and happens a lot, you collect the sample, you follow the protocol they followed in humans, which is like maybe flash freeze them or put it in a specific buffer, etc. But the transporting is not controlled as you would do it in the lab. So maybe even if I did everything right, it might still not be exactly to the standard that the sequencing facility or whoever is going to do the downstream stuff is expecting. So then I need to ask, okay, is it worth continuing if it's not up to human standards? And no one else has done it before. So it's like, okay, how do I decide if it's worth the money and the time and etc.? So that is challenging. Getting the samples, I think, is very challenging. Once we have the DNA and the sequences in the server, I think it gets less challenging. But the first, getting the materials, that's the first bottleneck in our research.
And so far you have decided that it is worth the time and money.
I think it's worth the time for sure, and the effort, because, at the end of the day, understanding evolution from the animals and the ecosystems out there is much closer to reality than trying to understand them if we have them in the lab in a controlled environment. So then our results are reflecting what is happening. It gives a better picture of evolution.
Another challenge is the lack of existing knowledge.
I am studying what we call non-model organisms, species that are not humans or not used to understand humans in a medical context or biomedical. So that means that most tools in genomics are made or are invented and perfected to work very well for humans. So human DNA or human RNA, they're optimized. All protocols are optimized for humans. So when I am trying to apply these same tools to my organisms, there's always some optimization that needs to be done, always some challenges that need to be overcome. So I guess in the day-to-day, okay, if I have this protocol and I want to use it for birds or mammals, I need to try it. It will likely fail a bunch of times before it works. And what you would do is you try to troubleshoot with some colleague that has done that before. If you're working with humans, there's probably many colleagues who have done that before. When you're working with the species I work with, there's not so many colleagues working with them. So sometimes we're a bit on our own trying to figure out what works.
So why not work with humans or model organisms?
I think about it all the time. Maybe I should just switch. But at the same time, it's like once you find your system that is teaching you a lot about evolution, and you can see the benefits of it. So for now, I think it is teaching us a lot. And I'm still trying to see what is the limit that I can do with this species that I'm studying. Also, if we want to understand, something separate from humans entirely, right, about what the ecosystem itself, how is it evolving? Of course, ideally, you would study all species that exist in this ecosystem. So the more we can study, the better, the more realistic picture of evolution we have. If we study all its components and all the species involved.
Cell lines are populations of cells kept in a lab. Since the group can't test all genetic hypotheses in the field or experiment on wild animals, they are now creating their own cell lines from the animals they study. They do this because existing cell lines from model species like humans have proven difficult to use in their research.
We collected hair samples from Norway and we isolated the cells from the hair skin, and now we are working on the downstream functional validation in our own hair cell line. So we really need to spend a lot of energy and time to optimize the experimental situation. Yeah.
So how did Mafalda end up studying color change?
I really wanted to be a field biologist and work in conservation and save the world. I wanted to work with lions.
Working with lions was hard to do in Portugal, but Mafalda knew that other researchers worked with the Iberian lynx.
Now it's doing much better, but at the time was the species that had a big conservation concern in the Iberian Peninsula. So there was a big effort towards conserving them. And I was fascinated by that. I really wanted help in some way. So it was closer to home. And so I was looking for a project where I could work with mammal species and do conservation. And I met this professor who surely steered me away but genetics is actually a really cool thing you can do for conservation. I was like, okay, that sounds interesting, but could I work with your Iberian lynx? That's what I was thinking about at the time. And he was like, well, maybe, but I have this project with color change. And I was somehow captivated by that, both by the color change, I thought it was an interesting system, but also by the possibility of going abroad, which was something that I really wanted. So instead of ending up in the south part of the Iberian Peninsula, I ended up in cold environments with lots of snow and negative temperatures. But it worked out. I'm happy. Yes.
And now that you've been a researcher for quite a while, what's it like?
Oof, it's a roller coaster, I would say. I have the privilege to focus on the things that I'm very excited about. And that I think are important. I think also that now that I've become a researcher, group leader, right, and I get the chance to build a team that is learning from my experience, and I'm learning from them as well. I think this person-to-person relationships that you get to establish in this job are, for me personally, very rewarding. And also the amount of people you just meet, right? I know people from all over the world. The colleague sitting next to me is from Argentina. The one in the office next is from Iran. Outside we have students from Sweden, from Greece, from all over Europe or even around the world.
But life as a researcher doesn't come without challenges.
Our research is only possible if someone is convinced that it's cool, right, and awards us funding. So that aspect sometimes can be very frightening, I think, and very stressful. And we're always facing challenges, right? It's a lot about being resilient and staying positive that we're doing great work and the best that we can, even though that many times we're faced with a lot of failure and things are just not working the way that we wanted, or we don't get the answer that we're hoping for. From the data we just collected, we spent I don't know how many days in the field sampling, and then it didn't work. But I would say I'm very grateful that I get to do this job. Yeah. Even if sometimes I would like to quit because it's so stressful.
How do you stay positive?
Well, we always have, among the failure and the challenges and etc., we do have some, there is something that comes in. There's results that we get that are really exciting, that, maybe at first they weren't what we are expecting, but then we're like, okay, that's okay, I learned something new. We do get funding every now and then, and also I do stay positive by having these conversations with colleagues about, I think it's really important, this community that you establish where you can, realize that, this is what it is for most of us. It will be okay in the future, right? Others have gone through the same struggles. It's kind of what it is, and things work out. But also I think I stay positive because I trust that being part of SciLife Lab and being in Sweden, for example, I know that people here really do trust science and value what we do. So that means, what I'm doing is relevant and important and valued. So I should keep going. Yeah, and I stay positive by also having time for myself and for the things that I like to do outside of work. So that puts things into perspective, right? It's not like, if things fail, it's not the end of the world, right? It's a job as well. This is my job, and I am other things as well that are important. And yeah, I don't know, I like to spend time with my husband, my family. So, all those other things are also just life and important to me. I do a lot of hiking, I like to climb, I do a lot of crafts, so I like to knit and crochet and embroid.
Have you made this one?
This one, yes, I did make this one. Maybe you can tell it's not very good. It's like, but yeah, I did make this one.
I didn't think it was homemade when I saw it. Please give me this one.
No, I made this one actually. Yeah.
That was very good.
Thank you. I could make my own clothes, so I'll be fine.
All right, what's next with your research, Mafalda? And what will happen to the species you study if their environment changes due to climate change?
Snow cover is declining, so it's not really good for them to be actually changing to a white winter color in the winter time. So can they stop changing color? And if they can, so some species can, so is that genetically determined or plastic? So more of a developmental control. So there's a lot of interesting evolution questions in the system that we can answer. What our research suggests is that if there is genetic variation, to a certain extent, ecosystems or species will be able to respond rapidly to climate change. And I think classically in evolution, we've thought evolution is slow and can take millions of years. But as we study these systems, we realize that evolutionary response can actually be quite fast. In a matter of 100 years, we predict that these animals, if there is genetic variation for color, for example, they should be able to adapt. Of course, it's not just about snow for these species. We see that if snow is gone, they could adapt, but it's not just snow, it's also habitat fragmentation, it's certain species, it's also over hunting, for example, or the invasion from other species that, other species are trying to find better environments in climate change, so they might move north because they're tracking the temperature. So then you have, an interaction. So it's very complex. But what our research suggests is that if there is diversity, and if we keep that diversity, that gives a better chance to species to adapt. I think that's a very positive outlook for the species we study and for other species. But there are caveats. And so I, as a scientist, always have to be like, I know there are caveats and I can't say they're going to be fine. The hope is that we can use more information, not just about genetic diversity, but also genetic diversity that we know matters for adaptation currently or into future environments to inform conservation.